Model 11 Numerical chromosomal aberrations, under that euploidy, monoploidy with suitable examples. The main objectives of this episode are to know about the numerical chromosomal aberration, to understand the type of numerical chromosomal aberrations, to explain about the euploidy condition in plants and animals, to know about the monoploidy condition in plants and animals, to know about the significance of euploidy and monoploidy conditions. Introduction. All eukaryotic organisms have their chromosome numbers which are different in number across the species. Each sexually reproductive organism consists of a diploid chromosome numbers in somatic or body cells and haploid chromosome numbers in the germ cells. The number of chromosome sets or copies in each cells is generally termed as diploidy. Usually, the eggs or sperms are said to be haploid by the presence of a single genome or haploid set of chromosome. After the fertilization of sperms and egg results in the diploid zygote, where two sets of genome are present that comes from father and mother. Some of the changes may occur in the chromosome and result in the changes in the number of chromosomes. The changes may occur during the cell division or by the result of radiation, chemicals, etc. The number of chromosomes varies in the cell and gives rise to many abnormalities. The change in the chromosome may lead to the structural aberration or the numerical aberrations. The changes in the number of entire chromosomes are termed as the heteroploidy. Heteroploidy is of two types, including changes in the whole set of the chromosome known as euploidy, and the changes in the chromosome numbers by addition or deletion of single chromosome results in the aneuploidy. In the case of euploidy, the entire set of the chromosome may gain in number or it may lose the whole set of chromosomes. In the aneuploidy set, as the whole chromosome along with the extra number of single chromosomes may present. Monoploid is the condition of the presence of a single set of chromosomes. Hence, in this episode, the following aspects are going to be discussed. 1. Numerical aberrations in chromosomes. 2. Euploidy condition. 3. Consequences of euploidy. 4. Monoploidy conditions and significance of euploidy and monoploidy. Numerical aberrations in chromosomes. The abnormality in the number and structure of the chromosome in any organism is called chromosomal aberrations. Depending on the type and the kind of the aberration, it can have severe birth anomalies which can lead to defects incompatible with life. Chromosomal aberrations can be identified and visualized with the use of a karyotype. Changes occur in the number of chromosomes by the deletion or addition is the result in the numerical aberrations. The numerical changes in the chromosomes in cells result in the different conditions of ploidy in humans. The defects in the chromosomal segregation during the reproductive cell divisions may result in the cells with an extra number of chromosomes or the loss of chromosome in other cells. At the same time, if the segregation defects occur in mitosis, results in the mosaic condition of aneuploidy. In many cases, the numerical changes result in the pregnancy loss or other abnormalities in the newborn baby. Usually, these numerical changes occur in the old or aged cells, where the improper cell division results in defects. Both numerical as well as the structural chromosomal aberrations are involved in the development of carcinogenesis. Importantly, the numerical aberrations are classified into two following types: euploidy and aneuploidy. Gaining or losing the entire set of chromosomes results in the euploidy condition. In a normal diploid condition, where 2n generally called as 2n, if one more complete set of the chromosome is added, then it is termed as the triploid 3n condition. Tetraploid 4n results from the addition of two entire sets of chromosomes to the diploid state. Gaining of the whole set of chromosomes in more than two sets is result in the polyploid condition with pentaploid and hexaploid, etc. Aneuploidy is the conditions of the presence of an abnormal number of chromosomes. There will be gain or loss of one chromosome instead of the entire set of chromosomes. Trisomy, that is 2n plus 1, is the condition where the normal diploid condition 2n with one extra chromosome is present. In the case of humans, diploid condition shows the 46 chromosomes with two haploid set of 23 chromosomes come from father and mother. In the case of trisomy conditions, they show 47 chromosomes. Monosomy 2n minus 1 is the condition of having a loss of one chromosome from the actual set of chromosomes. Monosomy condition in human shows 45 chromosomes. Euploidy condition. Increasing in the set of haploid chromosome number leads to the euploid condition. In the case of euploidy, the genome contains more than 2 copies of full haploid chromosome number. The number of chromosomes present in the normal basic set is generally termed as the monoploid number. It is designated as X. In the case of diploid condition, the monoploid number is 2x, and in the haploid condition, the monoploid number is 1x. In the case of polyploid condition, the monoploid number shows more than 2x. The number of chromosomes present in the gamete cells is generally called as the haploid number. In most of the diploid animals and some plants, the haploid number and the monoploid number will be the same. For example, in humans, the haploid number in gametes shows 23 chromosomes and the monoploid number 1x proves to be 23 chromosomes. In the case of a few plants, the monoploid number and the haploid number are different. For example, in the case of wheat with chromosome number 42, the haploid number in the gamete is found to be 21, whereas the study shows that it is hexaploid and hence 6x is equal to 42 chromosomes. The monoploid number for the wheat is found to be 7. C. Consequences of euploidy in animal and plants. Euploidy in animals. Increasing the number of chromosome set will lead to the polyploid condition. The polyploid condition is infrequent in the case of animals. Change in the number of chromosome will disturb the balance in the animal genome, and these variations can finally lead to the sterile condition. Only in rare cases, the animal can avoid the hazards of polyploidy. For example, in the case of brine shrimp, it utilizes the parthenogenesis to initiate embryo development. Parthenogenesis is the development of an organism from the egg without the fertilization. Euploidy in plants. Polyploidy can be easily tolerated in case of plants. The two major categories of polyploidy are autopolyploid and allopolyploid. Autopolyploid is the condition of having an additional set of a chromosome that is identical to the parental species. When we considered an example of two species, capital A equals small a 1 plus small a 2 plus small a 3 and plus and so on, where the individual chromosome is represented by A1, A2, etc., and n represents the haploid number of the chromosome. Another species, that is called capital B, equals small b 1 plus small b 2 plus small b 3 plus up to small b n, where b 1, b 2, etc. are the individual chromosomes and n is the haploid chromosome number. The diploid condition shows capital A chromosomal composition. In the case of autopolyploid condition, triple A, that is called autotriploid condition, where 4 A's called autotetraploid or the chromosomal composition. In case of the allopolyploid conditions, individual consist of an additional set of chromosomes that are derived from another species. When we consider two species A and B mentioned above, their doubling of chromosome number and their chromosomal composition will be AABB. The doubling of chromosome number will lead to the allotetraploid condition. The research on the allopolyploids was initiated in 1928 by Karpetchenko. The condition of autotriploid can be formed when the normal gamete having the haploid number of chromosome N unites with a gamete that has not undergone the reduction division that remains in diploid condition, that is 2n. Hence, the formed zygote will be of 3n. The triplets can also be formed by the mating of diploid with a tetraploid. The problems arise during the mating of autotriploids because of the pairing problems with additional chromosome set. The unbalanced gamete are produced. Thus, they become invariably sterile. In case of autotetraploid, except in some cases, they can undergo normal meiosis even with the additional set of chromosomes. Monoploidy conditions. Monoploidy condition results from the presence of one set or basic set of chromosome instead of two in normal diploid organisms. It can also be defined as the individual consists of one half of the normal number of chromosomes. Presence of only one set of chromosome is not enough for the development of diploid organisms. The allele show only one copy and hence masking of some of the lethal alleles is not possible. Therefore, in most of the animals, the monoploid condition is lethal. In the case of some animals, the monoploidy conditions can be occurring where the unfertilized egg can develop into an embryo. The process is called as the parthenogenesis. In the case of male bees, ants and wasps, they are the monoploids developed parthenogenetically from the unfertilized eggs. These are developed from the gametes which are formed by the mitosis because of the absence of pairing chromosome in meiosis. Hence, the meiosis will not occur and the monoploids will remain in sterile condition because of the meiotic problems. Usually, the monoploid conditions occur very rarely in the population as the aberration. In the absence of meiosis, the single set of chromosome will show random segregations. Monoploidy conditions in human show a single set of 23 chromosomes instead of 46 chromosomes. As a result of recessive lethal mutation, the development of monoploid fetus will not occur. They will die before birth. In normal cases, these recessive allele expressions are masked by the dominant alleles in the heterozygous conditions. Monoploids can be formed naturally in some animals and plants. They can also be produced artificially by the X-ray treatments, delayed pollinations, distant hybridization and production of monoploids from anther or pollen culture. Production of monoploids plays an essential role in plant breeding. Delayed pollination can be done by avoiding the normal pollinations, and which can make the development of egg parthenogenetically into monoploids. In the case of distance hybridization, the hybridization between two unrelated species is done. This intergeneric or interspecific hybridization will lead to the formation of monoploids. Anthers and the pollens are the reproductive part of the plants, where they are taken and grown on the medium with necessary nutrients, and they produce the haploid plants. Monoploid in animals and plants. In the case of diploid condition, the alleles for the recessive condition get masked by the dominant alleles. Hence, those recessive lethal alleles are not a problem in case of diploids. In case of the monoploid conditions, the recessive mutations get unmasked and the organism can die before they are detected. Some of the species such as bees, ants are monoploids. They develop from the unfertilized eggs and they remain sterile. Some of the fungal species also show the monoploid condition in their life cycle. Most of the sexually reproducing fungal species show haploid phase alternate with diploid phase. The haploid phase gets to end with the nuclear fusions and switch to the diploid phase, where the formation of zygote takes place, resulting in the formation of a diploid cells. Significance of euploidy and monoploidy. Aneuploidy or abnormal chromosome is common in cases of human also. Human embryos can have the abnormal chromosomes, and it leads to the miscarriages. It is also responsible for the development of congenital disabilities. These chromosomal aberrations are mainly because of the meiotic errors that occur during oocyte generation. These meiotic errors can increase with the increasing age of woman. As the woman gets older, the chances of meiotic errors or non-disjunction occur more often with old eggs. Monoploid is successfully applied in plants breeding techniques. The anthers from the plants are developed with fixed phenotype in plant biotechnology. Then by using tissue culture, the anther culture is used to regenerate new plants. Here the plants will remain in the monoploid conditions. The genetics of these plants can be studied, or chemicals can be used to double the chromosome number to form the diploid condition. Plants with desirable traits can be selected in the F2 generation by plant breeders. These lines will not be in a genetically homogeneous condition for that trait. Several more generations of testing should be done to get the desirable trait a fixed line. The chromosomes can be doubled using some drugs. The gametes get fixed because the individual will remain the homozygous conditions for every gene in the genome. Even though the plant biotechnology successfully used, the limiting factor is the development of anther culture techniques for different crops. To conclude, euploidy is the presence of additional chromosome sets. Monoploidy refers to the normal condition where one set of the chromosome is present. These euploidy and monoploidy conditions are found often and useful in the life cycle of plants and are very rare in the case of animals. Thank you.